I would like to talk about innovations in brewing raw materials and also how some of these innovations and findings and knowledge can be used to improve other crops in the world. When we talk brewing raw materials, barley, malting barley, is one of the key raw materials for beer brewing. On top of that, we also use water, of course, then there's yeast, and then there's hops as the key ingredients for beer production. Beer brewing is really based on high-quality raw materials. So malting barley has to consist of a very good quality, high starch content, low protein content, but also other desired properties, just like that it has a relatively low level of fibers and other things. So again, the beer brewing process can run very smoothly. We have for years been looking into many different steps of all the key raw materials for beer brewing and how we can optimize that. Beer brewing is a long process. It has many different steps. And again, here innovation is also very important for the future on how we can improve and how we can make beer brewing more sustainable, also from a raw material and crop point of view. So when we talk malting barley and improving malting barley, then it's also very important then to understand the key fundamentals. Everything from genetics, biochemistry, plant physiology, and also agronomy, and how that play into a plant that can produce a very good quality. Therefore, we have a lot of scientific data from many years of research and science that we can use today. And that combined also with the barley genome sequenced, which we published in 2017 as a part of a big international consortium, has really given us a fantastic new toolbox to improve barley, but not only barley, but also using these experiences and learnings from the barley genome to be used for improving other crops and for improved quality as well as productivity and better yields. The barley genome, when it was sequenced, has been a huge effort for the group here. As I said, it took more than 10 years of very hard work, sequencing work, assembling the whole genome, and getting the whole thing into the right order so it can be used scientifically. We have at the same time and in parallel also been working a lot on finding out what kind of genes are expressed, when are they expressed, what tissue are they expressed. That combined with the sequence and also with the scientific knowledge we have from the biochemical studies as well as physiology studies is now giving us a fantastic opportunity to not only use it for barley development and improvements, but also for other crops. We have also been looking a lot into how can we use this data from the genome, understanding the genes, in order to improve the quality of the barley in such a way that we can improve, first of all, taste, freshness. It could also be foam stability and other things. And today we have one world-leading pipeline on malting barley with new improved traits for the brewing and malting business. A few years ago, we started also to develop a new technology where we really could accelerate this development. And today that has more than 1,000-fold increased the efficiency to identify new genetic variants which we can use for improving our malting barley. So again, this is extremely valuable information that we also will be able to use in the future for barley, but also for other types of plants. So when we then continue to develop barley or other brewing raw materials, we use the conventional breeding methods. We are not using any GMO technologies, conventional breeding technologies where we are making crossings, we are testing, we are selecting, we are using genetic markers in order to really follow the different traits that we are interested in keeping in our pipeline. Many of these lines will, after selection, also be planted in fields. And here is one example here from Fynen in Denmark, where we have one of our bigger scientific research fields, where we are testing all the new malting barley varieties. And only a few of those lines, around 20 to 25% of them, will actually succeed and be selected for the next stages. So it's a very hard selection that's being performed in the whole breeding process. Today, it takes us approximately 3.5 to 4 years to make a new malting barley variety. Previously, it has taken 10 to 12 years, but now with many of the new technologies where we can accelerate, where we can have fast selection, and where we can also send barley to, for example, New Zealand, to the other side of the globe, we can have 2 generations of these new lines in a year, and that can help to speed up the whole production time of new varieties. Today, I think it's very pleasing to see because we have a long list of new traits from malting barley with improved freshness, quality, but also higher yields. And one of the newer things that will come up now very soon is that we will have new varieties that has an improved climate tolerance. So again, these will be barleys that can grow in areas of the world, for example, Australia and Asia and Russia, or Southern Europe, where there is heat and drought. And again, this is probably going to be a bigger problem in the future. We all hear about all the extreme weather conditions we are facing in the future. And again, for us, it's also very important that we can already now start that development to secure the future supply of the raw materials. We are also having new special barleys, red barley, black barley, that's being used for special types of beer production. And on top of that, also hops is a new research area, scientific area, where we again, from a very basic, fundamental scientific point of view, start to look at hops and all the fundamental processes in hops, hop development and hop physiology, as well as genetics, in order to understand hops and improve it exactly the same way as barley. So today, I think it's quite a promising pipeline we have with new climate-tolerant barleys. We also have a pipeline of new quality malting barleys, where we have improved the freshness of the beer, foam stability, but also the whole sustainability angle is now embedded in some of these new barley varieties, where we can save water and also save energy in the future in the whole production chain of beer production. Also, stabilization is a thing that's quite important when you want to make clear and filtered beer, that it has to be crystal clear. And here we have new barleys where we do not need to stabilize the beer. So again, now with having a strong pipeline of new malting barley varieties that can help to improve the beer brewing process, but also consumer experience in the future, we are very much engaged also with farmers, with grain companies, and with maltsters, because the whole value chain here is of key importance when we talk about getting these things into the market and also being scaled up in sufficient quantities so we can start to use it. And again, with the aspect of climate change, it's extremely important that all the members, all the partners in such a supply chain is fully on board. In the future, we also see that there is a need to continue the development of crops, not only barley, it could also be wheat, it could be rice and other things, in order to be able to cope with the climate change and the extreme weather conditions. So where do we get inspired from? Well, we have actually just started a bigger project together with Copenhagen University and with the help of Carlsberg Foundation, who is sponsoring the project. We will look into a new plant that has been, some years ago, been introduced into Europe. It's actually originating from South America, a plant called Chenopodium quinoa, which is extremely salt, heat, and also drought tolerant. And having such an extreme plant that can cope with all these things is of course also a valuable tool to use also for developing other new types of crops to gain a higher yield and better quality, as well as a better life for farmers around the world. So again, understanding the fundamentals of drought and heat tolerance and salt tolerance is something we hope we again can use and embed in our raw materials for the future. Another inspiration has also been another Carlsberg Foundation supported project, namely on barley domestication. So ancient DNA, we call it, because here we will sequence very, very old barley grains from the old Egypt, around 3,500 years old, which we were so lucky that we could get a few of from the Glyptotek here in Copenhagen. It was found in one of the old Egyptian graves, and now having these grains in our hands and getting the DNA, this ancient DNA, extracted out of those, we will hopefully be able to compare those with modern barley varieties and see during those studies if traits, if properties has been lost during this domestication. And hopefully there will be a chance, if there will be some valuable traits, that we can reintroduce them or in modern crops in the future. A similar study has also been initiated together with Eske Willerslev, and that's relating to rice domestication. So here, samples will be taken out from many different rice paddocks in China. Very old and ancient rice samples will again be tested and will be analyzed, and hopefully we can also extract DNA out of those samples and compare those with modern rice varieties in order again to see if there will be traits that has been lost during domestication and could be of value today and for the future. And finally, hops. We, as I said, have developed quite a lot of tools and new ways of redesigning and adapting the crops and the raw materials we are using for improving and making consumer benefits. And here hops is again one of the key raw materials for brewing. And again, hops was actually given up here in Scandinavia and Denmark back in the 50s. We have 2 years ago or 3 years ago started to reintroduce hops in Denmark, and again using the same approach to adapt it and find the best suited varieties and lines here for Scandinavia as well as for Russia. And as a part of this study, we will also sequence the hop genome. Back in time, we sequenced the yeast, a part of the yeast genome, then the barley genome in 2017, and hopefully now also soon we'll have the hop genome sequenced. So finally, I think this was a little insight to how barley could be used as a model organism, also to help to improve other crops for the future, for higher yields, for better quality, for the world population. Thank you.